My name is Ignacio Larrabide, and I will present to you the latest developments of our group in Voxel-Based morphometry. So quickly, an agenda for this presentation is I will start by showing you some results on the value of BBM in the lab, where basically we try to assess in a more quantitative manner how sensible BBM is when we are looking on or when we are working on animal models. Then I will transition to a more clinically oriented problem where we are interested in assessing BBM value in a clinical setup, where we need to deal with individuals instead of a group. So the first part of this talk is called the BBM sensitivity to localization and extension of mouse brain lesions with the simulation approach. This is a work that has been done together between our group in Pladema Institute, with ENIS in Hospital El Cruce, and the University of Calgary, who actually acquired the data. This is somehow the seminal work of Delfina Bragio. She's a PhD student in our group. You have her email in this slide to contact her in case you have interest or more questions about this paper. So basically what we have done here is that we have tried to assess the limitations that we have using BBM on animal models. Animal models have some limitations, like the size and the topology of the brain tissue is not the same as in humans. Sample sizes typically are smaller, so we have a lower N in this case. And the image resolution also changes, not only in the term of the number of voxels or pixels in an image, also in the size of each one of them. So we have to be very careful on how to process and we prepare the pipeline to work with this data when we are we need to check for the sensibility of this process. So for you who are not familiar with it, voxel-based morphometry is a statistical technique for neuroimaging that allows detecting and quantifying local tissue differences between two groups. Okay, so these two groups of individuals are mapped into the same coordinate space, and the tissue densities for cerebrospinal fluid, white matter, and gray matter are set on the same spatial coordinates. And then we compare voxel-wise the differences between these tissues across the groups. So animal studies, in animal studies, there is no consensus on if the pre-processing workflow or the proper validation haven't been done so far. So in our study, we proposed a simulation-based validation. So this simulation of atrophy is possible with simulation of atrophy is possible to have the proper ground truth, so we can actually identify very, very accurately where the atrophy is, which in real biological samples is not possible, even with images or even by a destructive analysis of the tissue. Two different pre-processing workflows were used, okay, and we'll go into detail of that later. And so the idea was to quantitatively assess how the results of comparing these two groups between the ground truth and the BBM results would come out. So first we did simulation of the atrophies. So different sizes and intensities of atrophy were applied. For this we used the work of Carasalis, who actually provides a code for simulating these kind of atrophies from MRI images. So you have to do segmentation first, and then you kind of apply this atrophy using the original image and the segmented gray matter tissue. And then you get a new image basically with an embedded atrophy in the region that you decide. So you have to choose radius of the atrophy and the intensity of the atrophy, and then you can simulate how this would behave, how the image would look like. In this way, we can very accurately quantify how this atrophy acts. So we know exactly which were the voxels because we have the original image and the simulated image, and on that we can very accurately check what are actually the differences and measure what was actually BBM capable of finding from these modifications. So for all of this, we need to follow a workflow. So the workflow for BBM, the pipeline for BBM is pretty standard. It has been well defined. So there are a series of steps that you have to do. First, manually align the images, then a linear registration where you basically take everything to the same size. And then you have to register, so do an actual deformation on the geometries to match them on the same spatial coordinate. So in this case, what we have done is basically we created a second group from an additional set of 10 mice. We created a simulated group, which we had applied different types and different locations of atrophies in each one of them. And we used then 2 different workflows to register them on the same coordinate space. The first one is what is called the optimized BBM, okay, where only the gray matter segments are normalized, not the whole brain. This was proposed by Good et al. in Neuroimage in 2001. And the second pipeline that we tried was Dartel-based. So Dartel is an alternative segmentation method that normalization method, that basically improves the inter-subject normalization. So it makes sure that overall the segmentation is more coherent across the whole group. So in these 2 workflows, there are a lot of somehow decisions. These are pretty standard workflows, but there are some decisions related to the amount of smoothing and the how strong this normalization is, which might affect the results in the end. So we wanted to have this comparison in a more detailed way such that we can quantify if what we observe in a real study where we don't have a ground truth is actually due to the normalization itself or is more due to actual differences between the groups. So in this next slide, basically we show some results where we compared the precision of the method. So this is somehow if the method is finding what is not real. So how good is the method to identify real atrophies, okay? And the recall, which is how much the method is actually missing atrophies. So do we have atrophies that the method is overlooking? And in general, we found out that Dartel did a much better in terms of precision, okay? But the optimized version had a better recall in the cortex. So we can observe here that the cortex itself, so the optimized method had done a better result, a better performance than that. Although overall, the Dartel method was capable of finding more atrophies in the whole set. Most importantly, both methods performed worst on the cerebellum. So the cerebellum is the region that the method actually showed problems to properly identify, and this is in our perspective is because the cerebellum has more variability in the tissue density for the gray matter, and this makes it difficult with such a small sample for finding properly these differences. What we then did was assessed this methodology in a real study. So in a study where actually we had 10 mice that were controlled, so they were normal standard. They haven't been altered in any way. And then we have 7 subjects with suffer maternal undernutrition during the pregnancy and the lactation. Okay, so after the comparison between these 2 groups, we had these results showing that the optimized workflow identified less of the regions with differences. Of course, here, as we don't have a ground truth, we have to somehow trust, okay, on what we have seen before, knowing that these are true findings and not spurious ones, okay? And this is somehow the beauty or the benefit of having a simulation approach, okay, that we can actually see exactly what we are doing and how things change from one group to the other, okay? Dartel somehow was more sensitive to the alteration in gray matter, showing that more findings under the undernurtured group, as you can see in this part of the plot. To conclude this first part of the presentation, pipeline affects the results. So there is an influence on how we pre-process the data and how the results will look like. Dartel had a better precision but both workflows underestimated some of the small differences, so it was not capable of finding everything. And in summary, the lower performance was observed in the cerebellum. In the second part of the talk, I will show you some results on using machine learning for classifying different findings when we use single-subject voxel-based morphometry. The main difference between classic BBM and single-subject BBM is that in the single-subject version, we are capable of assessing differences by individual. So this is good because it gives us a tool for assessing one individual and comparing him to a group of, for instance, healthy individuals. The main problem of this approach is that the number of false positives increase considerably when you do this kind of analysis. In this work, we also used a simulation of atrophy to introduce machine learning to find false positive clusters. This is part of the work developed by Hernan Kuzhar as his PhD in our lab. So when single-subject BBM is applied, a number of clusters is detected, no? But not all of these are real atrophies, because an individual can have normal differences with a group, and which are just a matter of differences, of natural differences between individuals. So this is why it introduces a lot of noise. So now what we could do was artificially introduce atrophies in visions of healthy patients, and then we kind of search for these atrophies to see if these were actually good or bad ones. And then we use a machine learning approach to classify these atrophies and see if we could filter them out when they were not real atrophies. So this machine learning approach, basically what we do is training a support vector machine model with the features, no? So these are characteristics of the data that are real and false clusters detected can have after we do single subject BBM. So we can in principle discriminate between true atrophies and false atrophies when a new case comes in. Support vector machines is a machine learning technique that can learn how the characteristics of samples from a given type. Okay, so from two different groups that will have these that we are interested in classifying. In this case, they are the true atrophies, which we call AT, and the non-atrophies, which we call NAT. Okay, so in this way, each sample will have a set of features, a vector of features that describes this particular sample. Then what we will do is these samples basically define a multidimensional space where all these subjects will live. Okay, so what we will do then is to find a function. This is called an hyperplane that best separates between the two groups. Okay, so these are the AT, so the atrophies and the non-atrophies in this group. This whole process is a mathematical process driven by optimization, and it's called the training of the support vector machine algorithm. The process of training runs on a series of features or characteristics that describe each one of the individuals that we want to classify. This is just a small set of features that we choose to use in this paper, but this can this is a whole area of research. So many different features could be used or could be found to classify these clusters, and I'm not going to enter into the details of this for the moment. For this, we use the ICSI dataset, so you have the link at the bottom of the slide. So we had 10 normal and healthy subjects from this group, which was quite balanced in the number of females and males, and the age was adults, healthy adults without any known pathologies. And we used the T1 weighted MRI images. To simulate the atrophies, we chose 3 different locations, trying to resemble a specific disease, like the hippocampus, the parietal lobe, and the superior frontal lobe. This is because we are interested in Alzheimer's disease, in temporal medial lobe epilepsy, and frontotemporal lobar degeneration. And we chose to do atrophies of different radii. So this was 20, 30, and 40 millimeters in radius. So here are some arrows that you can basically see how, as we increase the size of the atrophy, this goes from left to right, increasing the size of the atrophy, we can see how the atrophy actually alters the tissue and enlarges some of the voids, let's say, of gray matter in these images. For this, the Carasali code was used. We defined a series of different models of different sizes. So we we trained models using the same size of atrophy in some cases, and also mixing the sizes of the atrophies. This was a part of the experiment where we actually wanted to see if there was a size of atrophy that was ideal of clusters found in that atrophy, that was ideal to have a best performance in the model. And then we have models with single sizes of atrophies and models with a mix of all of them. So these were called basically SS for single size and MS for multiple size. And in total, we had 12 models, so 3 different locations. Each model was trained with the groups of clusters from the corresponding anatomical region. And then we came to the validation, to the assessment of the method. So as we can see in this plot here, we see that in gray is highlighted the number of clusters found for each one of the subjects. Okay, so each point in this plot represents a subject and the number of findings in that subject, and we are plotting here the non-atrophy cluster. So the larger this bar is, it means that more false positives were found on each one of the subjects. Okay, we see here that by a large different single subject, BBM was finding a lot of false positives, that after applying the filtering by support vector machines, we can see this model, this value going down to actually less than just a percentage of that. But this is not all of it, because we want to make sure that not only we are eliminating false negatives, but we are keeping the true positives, right? So we have to make sure that we remove what is not an atrophy, but we keep what is truly an atrophy. This is shown in this next slide, where we can see exactly that. So we see in gray the actual true positives detected by single subject BBM, and we see that after each one of the filterings, so in this case the 20, 30, 40 or multiple size models, that we preserve this. So what we observe here in general lines is that there is little to no changes in the number of true positives found in the case, which is actually good news. These are rock curves for the different regions, so we can see for each one of the colors one of the models and how it performed with the corresponding test group. We see a similar behavior for the parietal lobe and also for the superior frontal gyrus. So finally, one of perhaps the most valuable points of BBM is that not only provides a classification, yes or no, true or false, it also provides a score that can help or can be used during the diagnosis or during the clinical stage to help us knowing which are the clusters that we are not certain whether they are or not real atrophies. Okay, so it's not black or white. It can give also a score, giving us the certainty that the model has on this particular classification. As conclusions, we can see that there was very similar atrophy regions between detections between different sizes, and the performance of the model was actually similar, but let's say the model that has multiple sizes can have a better performance overall. all the non-atrophy clusters, SVM is a useful tool to identify doubtful classifications. Okay, so this is then the end of my talk, and I would like to thank you all for presenting this. This is just the faces behind the work. Thank you very much for everyone.